Good morning, students. This is Dr. Pratibha K.S., Head Department of Biotechnology at KLE SNC. Today, I am presenting before you important questions from Paper 7, Plant Biotechnology. I hope you make use of today's list of questions. I request you to invest your quality time and, if possible, try to answer the given questions. If in case you're not getting answers for some of the questions, you can reach to me through comment box. Alright, as usual, before going through today's class, let's have thought for the day. Be thankful for everything that happens in your life. It is all an experience, right? ಮತ್ತೊಂದು ಅನಗತ್ಯವಾದದ್ದನ್ನು ಹೊರಗೆ ಹಾಕಿದಾಗ ಮಾತ್ರ ಅಗತ್ಯವಾದುದಕ್ಕೆ ಜಾಗ ತೆರೆಯುತ್ತದೆ ಅಲ್ವಾ? ಮನಸ್ಸಿನಲ್ಲೂ ಅಷ್ಟೇ, ಮನೆಯಲ್ಲೂ ಅಷ್ಟೇ. ಆದಷ್ಟು ಅನಗತ್ಯವಾದದ್ದನ್ನು ನೀವು ಹೊರಗೆ ಹಾಕ್ತಾ ಇರಿ ಅಥವಾ ಅನಗತ್ಯವಾದುದರಿಂದ ಆದಷ್ಟು ನೀವು ದೂರವಾಗಿರಿ. ಓಕೆ, ಐ ಹೋಪ್ ಯು ಅಂಡರ್ಸ್ಟುಡ್. ಥ್ಯಾಂಕ್ ಯು. Now let's begin with today's list of important questions from Plant Biotechnology. Here I am dealing 2 marks, 5 mark, 10 marks and 1 mark questions. Okay. Now let me go through the first one, 2 mark questions. First one is totipotency, somatic embryogenesis, organ culture, protoplast culture, acclimatization or hardening. List the surface stabilizing agents, parthenocarpy, embryo rescue, phytoalexins, callus, explant, pistil. List the different plant tissue culture. What is hypochlorite dedifferentiation? Students, I've taken most of the questions by referring university question papers. So, kindly value all these questions and try to answer the questions. Okay. Further, what is acetosyringone, mericlones, vegetative propagation, reporter genes, endosperm culture, subculture, what are fusogens? Name 2 criteria for plant variety to be recognized under PBR. List the types of culture medium. Okay, you can take screenshot and you can go through this particular questions. Okay, next moving on to 5 mark questions. First one, briefly explain surface sterilization of explants. Discuss in vitro methods in plant tissue culture. Briefly explain aseptic techniques. What is patent? Mention its applications. Explain shoot tip and callus culture. Write a note on applications of micropropagation. 7th one, what are protoplast? Explain isolation of protoplast from leaf. Autoclaving, plant cooperation treaty. So these are some of the important 5 mark questions. Okay. Now let's move on to 10 mark questions. In this first question, what are secondary metabolites? Explain their in vitro production. Second question, write short notes on auxins, cytokinins and gibberellins. Third question, describe the methods of isolation of protoplast. Fourth one, define plant tissue culture. Discuss any one method of inoculation of explant. What are growth regulators? Discuss any 2 hormones. See, I have asked here what are growth regulators. Discuss any 2 hormones. I have asked here to write only 2 hormones. Here you are writing on auxins, cytokinins and gibberellins. Okay. Next question here. Define plant tissue culture medium. Add a note on plant tissue culture media. 7th question. Describe the stages involved in micropropagation. Discuss in detail about somatic embryogenesis. 9th question. Discuss basic requirements and techniques of plant tissue culture laboratory. So these are some of the important questions according to so far what are we have covered plant biotechnology unit. Now let's move on to 1 mark questions. Now, first one is name the instrument used for dry heat sterilization. Students, you will get answer for all these questions in my previous YouTube channel, that is Plant Biotechnology question and answers. Okay. What is culture room? What is electrofusion? What is totipotency? What are growth regulators? What are the explants used to haploid production? What are synthetic seeds? Write an application of plant tissue culture in forestry. What are biotic elicitors? Expand PGR, TRIPS, GATT, IPR, LAF. Kindly pause the video. Okay, then you can make a note of all these questions or take screenshot. What do you mean by explant? Further, define patent, define trade secrets, define plant breeder's right, define copyright, define callus, name two cytokinins, what is stepatum, what are hybrids, what are edible vaccines, what is endosperm, list the applications of plant tissue culture. Okay. Further, moving on to 22nd question. What are transgenic plants? Haberland, hairy root culture, acclimatization, soma clones. Name any two commonly used vitamins in tissue culture media. Expand BAP. What are somatic hybrids? What is fluorescent diacetate? Okay. Further, moving on to 30th question. What is regeneration? Potato late blight is caused by? Which explant was used for propagation of orchid plant Noacia nidus avius? Expand HEPA. Name the fungus which gibberellic acid was discovered. What are heterokaryons? Name the disease caused by Agrobacterium tumefaciens. Next one is define embryo culture. 38th question. Define organogenesis. What is protoplast? Define sterilization. What is cell suspension culture? Expand IAA, BAP, NAA to 4D. List out the surface sterilizing chemicals used in plant tissue culture experiments. Next, name two stains used to check viability of protoplasts. What are the chemicals used for protoplast fusion? Name the two chemicals required for the preparation of synthetic seeds. What is hybridization? Okay, so these are some of the important questions which comes in your plant biotechnology unit test paper according to so far whatever we have explained. Okay, I hope made crystal clear about the important questions. Thank you everyone for investing your valuable time. This is Dr. Pratibha KS signing out for the day. Stay tuned with me, stay safe and stay healthy. Thank you.